In this third installment of our discussion of the respiratory system from chapter 13 of our physiology textbook, we'll look at alveolar gas composition, hemoglobin saturation curve, and gas transport. How are the gases actually physically transported in the blood? So first, let's look at the gases in the alveoli. That's what the blood is going to encounter when it enters the lungs. So in the alveoli, the partial pressure of oxygen is around 104 millimeters of mercury. That's going to be the driving force, as we'll see, for diffusion of oxygen. However, we said that the oxygen partial pressure in the atmosphere at sea level is about 159 millimeters of mercury. So why so little in the alveolus? Well, there's three kind of contributing factors. Remember, we talked about anatomical dead space. When we inspire some air, the first thing that happens is about 150 mils of old air, spent air, that's in the airways, goes right down into the alveoli, and then we bring in some fresh air on top of it. They mix together and dilute some of the oxygen that's in the fresh air. Secondly, we talked about the nasal cavity, how it humidifies the air as it comes in, and it turns out that if you add another gas to the mixture, it will essentially displace the other gases. Why? Because the total pressure is always 760 millimeters, or whatever the atmospheric pressure is at a particular point in elevation and on a particular day. So all the gases contribute to that 760 millimeters. If you add another gas, all the other gases are going to have a lesser partial pressure. They'll have the same percent of the composition, but they'll have less partial pressure. So adding 47 millimeters of mercury water vapor, which is the water vapor pressure at saturation at 37 degrees Celsius, now we displace some of the oxygen. Diffusion, there's always oxygen diffusing out of the alveolus at all times. So as it's coming in, it's already starting to diffuse away. So anyway, on average, the composition of the alveoli is 104 millimeters of mercury in partial pressure. CO2, the alveolar partial pressure of CO2 is 40 millimeters of mercury. That's a regulated variable, as we'll see later, but just so you know, but it's 40 millimeters of mercury. CO2 is constantly diffusing into the alveoli and then being breathed out. But on average, that's our alveolar CO2 partial pressure. So, a little conflagration in the studio. All right, so here we see the alveolus. This is the alveolus. The air is out here. The partial pressure of oxygen is around 159. Not much CO2 at all. Air comes in. We deplete some of the oxygen or diminish the oxygen. 104, 105 millimeters of mercury partial pressure of oxygen in the alveolus. 40 millimeters of mercury CO2 partial pressure all the time. So that's what the blood is going to encounter when it comes into the lungs, and we're going to have the blood equilibrate or come to match the partial pressures of these gases as it passes through. In other words, we're going to get rid of some of this incoming CO2. It's coming in at 46 millimeters of mercury partial pressure, and when the blood goes out, it has 40 millimeters of mercury CO2 partial pressure. It's equilibrated or matched to the alveolus. That's a regulated variable, as we'll see again later. And we have some oxygen diffusing from the alveolus into the blood. There's a big difference in partial pressure driving that diffusion, right? 105 millimeters in the mercury in the alveolus, only 40 in the blood coming into the lungs at rest. And that drives oxygen into the blood. And once the blood has passed through the lungs, it will be equilibrated or matching the partial pressure of the alveolus, 100 millimeters of mercury partial pressure. And then the blood will be sent to the systemic circulation. Well, why is it that such a shallow CO2 gradient, only 45 millimeters of mercury or 46 coming into the lungs in the venous blood and 40 in the alveolus, we can still diffuse just as much gas of CO2 into the alveolus as we can diffuse oxygen out with a gradient of, say, 100 versus 40 of oxygen partial pressure. That's because, to remind you, we talked about this once before, CO2 is 20 times more soluble than O2. So we get just as much gas exchange with that shallow gradient for CO2. the alveolar concentrations don't necessarily remain the same all the time. Those values that we gave, 104, 105 millimeters of mercury, partial pressure of oxygen, 40 millimeters of mercury CO2, those are average resting values. It depends on the O2 absorption into the blood, and it depends on the rate of CO2 excretion into the alveolus, but it also depends on the ventilation rate. If we increase the ventilation rate, the alveolar composition starts to look more and more like the outside air. Right? The atmospheric air has 159 millimeters of mercury, so if we start to increase ventilation, let's look what happens to PO2. Here's our normal ventilation rate, where these two black dots are. Right? Ventilation is on the x-axis. So 4 liters per minute is our average ventilation. At that is the conditions where we saw 104, 105 millimeters of mercury partial pressure of oxygen in the alveolus, 40 millimeters of mercury CO2 partial pressure in the alveolus. But if we were to increase the ventilation rate, let's see what would happen. If we move to the right and ventilate more liters per minute of air, the CO2 partial pressure is going to go down, right? Because we're blowing off, it said, we're getting rid of more CO2 out into the atmosphere, taking in more fresh air that has no CO2. Oxygen partial pressure will increase because we're bringing in more of that higher partial pressure air from the atmosphere that's at 159 millimeters of mercury, and raising the alveolar oxygen content. So those numbers we gave were just the average values for resting conditions, but it definitely varies with ventilation. What if something was inhibiting ventilation? The CO2 will start to accumulate. If we start moving lower in liters per minute, more CO2 is going to start accumulating in the blood and in the alveoli, and there's going to be less oxygen because it's diffusing away and we're not bringing it in quickly enough. So increasing ventilation increases PO2 and decreases PCO2. Decreasing ventilation decreases PO2 and increases PCO2. There's terms for that. Ventilation is regulated such that the partial pressure of CO2 in the arterial blood in the alveoli is 40 millimeters of mercury. If it deviates from that, we will modify the ventilation due to chemoreceptor activity so that it always stays at 40 millimeters of mercury. Resting, exercise, you name it, this is the normal resting CO2 in the arterial blood and the alveoli. If we are ventilating in such a way that the CO2 is going down, right, below this 40, that's called hyperventilation. If we're ventilating in such a way that we're ventilating more slowly than normal, and the CO2 level starts to rise above 40 millimeters of mercury, that's called hypoventilation. So if I ask you what those mean, don't say, oh, ventilating fast or breathing fast, or say specifically, hyperventilation is when the PCO2 drops below 40 millimeters of mercury. We're ventilating extra. Hypoventilation is when the PCO2 rises above 40 millimeters of mercury because we're slowing things down. All right. In this graph, we're looking at the pulmonary capillary PO2. As the blood is flowing past an alveolus, and we know that it's going to equilibrate that blood with the alveolar PO2. Question is, at what point along the capillary is that taking place? If 100% represents the end where blood cells are going to go into a larger vessel away from the alveolus, right? Here's where the blood cells enter the alveolus, or enter the capillary of the alveolus. Here's where they exit. That's the amount of time we have, basically, or the distance we have to get the job done of getting oxygen to the blood. After only about 1/5 or 1/4 of the way through that capillary, we've already equilibrated the blood with oxygen in the alveolus. The blood is already at 104 millimeters of mercury or so, a partial pressure of oxygen. So we have all this extra reserve. What if we started cranking up the cardiac output, right? We started exercising. Now the blood's moving through there faster. It's probably going to take a little more of this overall path of the capillary to get things done, to get the blood equilibrated, but we still have tons of reserve. Maybe out here, well, at the 60% of the capillary length, we'll reach equilibrium. That's fine. So we have lots of reserve. That's the take-home message from this slide. What if the lungs weren't healthy, though? What if the respiratory membrane wasn't healthy? In a diseased lung, we're seeing that we may never equilibrate the blood with the alveolus before the blood goes out and back into the heart and through the pulmonary veins. If you have emphysema, you are never going to reach the appropriate partial pressure of oxygen in the blood. You just can't get enough fresh air into the lungs and CO2 out, and so you always have hypoxia, not enough oxygen in the blood. All right, so then we take that blood that has equilibrated with the alveoli, send it over to the tissues, and when it comes to the tissues, let's see what the partial pressures are there. In the tissue, the partial pressure of oxygen is only 40 millimeters of mercury, 100 in the arterial blood coming in, right, because it's matching the alveolus. That's a big difference. 100 versus 40 millimeters, that's the gradient or driving force for oxygen to diffuse out of the blood into the tissues, into the interstitial fluid, and over to the cells. What about CO2? In the tissues, there's 46 millimeters, 45 or 46 millimeters mercury partial pressure. And in the blood, there's 40, because that's what the alveoli have. That's the difference. That 46 versus 40 is the partial pressure difference, the driving force for diffusion of CO2 into the blood. And as you can see, the blood, as it leaves the tissues, leaves the capillary beds, it's exactly equilibrated with the interstitial fluid in the capillary bed. So it's really easy to remember these numbers if you just remember that. The alveolus, the two numbers you need to know, oxygen and CO2, are essentially the same as arterial blood. And then when we get to the tissues, the partial pressures in the tissues are the same as the venous blood, quote unquote, coming back to the heart from the tissue. So there's just those two pairs of numbers you need to know, and there's because that equilibration is taking place. Okay, let's talk about oxygen transport in the blood. How do we do it? Well, here's a hemoglobin complex. There are 4 peptide chains drawn like these little taffies. There's 4 of them, 2 alpha chains and 2 beta chains or peptides. So 4 separate peptides that kind of just attach to each other spontaneously. It's just how their amino acid composition is. They stick to each other. And in each one of those globin chains is a heme group, this is called. It's this heterocyclic ring. It's kind of a flat disc, organic chemical disc. And there they sit in each of the 4 globin chains. And in the center of each one of those hemes is an iron atom. And the iron atom in the +2 oxidation state can bind oxygen. So we have 4 binding sites for oxygen in every hemoglobin molecule or complex. And that's just yet another representation. These little corners, when you draw an organic chemical like this, those corners represent carbons, and here they've been drawn in for not every case, but more of them have drawn in, so you can kind of see the chemical nature of this thing. So that's just another globin or representation of a heme disc, I mean. We call it saturation. When all 4 of these hemes have an oxygen bound to them, then the hemoglobin is saturated. And that's what must happen as the blood passes through the lungs. We must saturate the hemoglobin with oxygen so that it has all the oxygen it can possibly carry, and it's on its way out to the tissues. Now here's something kind of borderline miraculous about hemoglobin. Its properties are very, very interesting. And the properties of hemoglobin with respect to binding oxygen are what allow us to live the way we live. Let's try to take a look and understand some of the features of this hemoglobin oxygen dissociation curve. If we take some hemoglobin and stick it in a container, remember that cylinder way back when we were talking about Boyle's law? Let's stick some hemoglobin in a container like that, and then we'll stick some oxygen in there under pressure. And the pressure in there, the oxygens are banging all over the place, is going to drive them onto the hemoglobin because it has a high affinity for oxygen at those 4 sites. And at 100 millimeters of mercury partial pressure of O2, if we stick that in the container, that's exactly how things are in the lungs as the blood passes by the alveoli. What percentage of hemoglobin is saturated with oxygen under those conditions in the lungs? 100%. On the y-axis, we have hemoglobin saturation. That means that all of the hemoglobin has 4 oxygens per hemoglobin, and we're saturated. Let's say we dropped down for some reason, we climbed up on a high mountain, relatively high mountain, and the oxygen level in the atmosphere started to go down, and we dropped down to only 80 millimeters of mercury partial pressure in the in the alveoli and in the arterial blood. Take a look at the saturation now. 99%, 98% saturated. So the hemoglobin has a high enough affinity that we have a ton of reserve in terms of what we can tolerate in terms of oxygen partial pressure in the alveoli, which we're talking about oftentimes partial pressure in the atmosphere. So we can occupy a very high altitude without any trouble because the hemoglobin remains pretty saturated until we get way up in the high altitudes. And then we maybe start getting ourselves into trouble. Let's see what happens if we carry the hemoglobin out to where the tissues are, or our cylinder, in our little cylinder, we deflate it a bit so that the oxygen partial pressure is the same as in the tissues, 40 millimeters of mercury. So it's like carrying the hemoglobin with its oxygen over the tissues. What happens? Some oxygen comes off the hemoglobin. It's no longer saturated. This little line right here represents the amount of oxygen that was liberated into the tissues from the hemoglobin as we pass through the tissues at rest. Typically, the tissues have 40 millimeters of mercury partial pressure of oxygen, or of oxygen, and therefore we're going to drop down to about 75% saturation. So hopefully you see immediately, wow, at rest in the tissues, we get rid of 25% of the oxygen. We still have 75% of the oxygen in reserve on the hemoglobin, and that's absolutely true. Another thing to learn about this hemoglobin saturation curve is that as oxygen binds to hemoglobin, first one, then another, the affinity for the oxygen starts to rise. And we pack that hemoglobin up with oxygen. It really sticks on there. Then we carry that oxygen over to the tissues, and once when oxygen pops off the hemoglobin, the affinity for the other one starts to drop, and then another one comes off, the affinity drops even more. And that's partly what creates this steep dissociation curve. The change in affinity of hemoglobin for oxygen. Isn't that wild? It's called an allosteric effect, when binding of something in one place on a protein causes a change in properties somewhere else. When I say protein, I'm talking about the protein complex of hemoglobin. So we have oxygen coming in from the fresh air into the alveolus, dissolving in the water on the surface of the alveolus, diffusing over into the capillary, into the red blood cell, and then binding to the hemoglobin. And so it sticks avidly onto the hemoglobin. We have saturated hemoglobin. Then we deliver, or we circulate or transport that blood over the tissues. And now the partial pressure around here is lower, and some oxygen starts to leave the hemoglobin and diffuse over into the interstitial fluid and into the cells for the use in their mitochondria. And as the oxygen starts to come off, the affinity starts to decrease, and about 25% of the oxygen comes off those red blood cells before they go back into a vein and back to the heart. I got a little overlapping there of the picture. Okay, so once again, at rest, the tissues have 40 millimeters of mercury partial pressure oxygen. If we deliver the blood to that location, about 25% of the oxygen comes off the hemoglobin and is available for the cells. And that's all they need. What if we start exercising, though? Then the mitochondria start grabbing a lot more oxygen. The partial pressure in the tissue starts to go down. Look what it is in this illustration. It's maybe only around 15 millimeters of mercury partial pressure. Okay, so all we did is drop from 40 down to, say, 15, and look how much oxygen dissociated from the hemoglobin. 50% or more of the oxygen just came tumbling out of the hemoglobin as we moved from 40 down to 15. Originally moved from 100 millimeters of mercury all the way down to 40, and all we did is release 25% of the oxygen. Now we're going to start exercising and move the partial pressure from 40 down to 15, a much smaller drop in partial pressure, but a huge release of oxygen. That's because of that change in affinity of the hemoglobin for oxygen as more and more oxygens come off the hemoglobin. And that is what allows us to live our lives. We can have this tremendous reserve available oxygen in the blood that can then be delivered easily to the tissues because all we have to do is start taking it. The cells start extracting it, and it just starts coming. It starts coming off the hemoglobin, so it's just the most cool thing about it. There's some other things that help us get the oxygen off the hemoglobin in the tissues. right? Because we got this system that's very good at loading oxygen onto hemoglobin in the lungs. It's high affinity, that's great. What about in the tissues? We need to get the oxygen off again, and there's three variables or conditions that are going to help us get that oxygen off in the tissues, even though we can load it very efficiently in the lungs. One thing that's happening is a chemical called bisphosphoglycerate that starts to accumulate in the red blood cells. As the blood flows to the tissues from the lungs, it's much warmer in the tissues, right? Deep in the organs and the capillary beds. There's a lot of heat being produced as we oxidize fuel molecules and hydrolyze ATPs. 2/3 of the energy involved is released as heat. Heats up the red blood cells, and so everything just happens faster. All the enzymes work faster, and we form more of this bisphosphoglycerate. Well, bisphosphoglycerate reduces hemoglobin's affinity for oxygen, so that helps us drive the oxygen off the hemoglobin into the tissues. That's partly why that 25% comes off at rest, and why even more comes off later on when we exercise. So we have that bisphosphoglycerate accumulating. The temperature itself, as we go into the tissues, the temperature is rising. Here's a little graph showing the hemoglobin dissociation curve of oxygen at 37 degrees. What if we go into the tissues where it's actually even warmer deep in the tissues, might be as high as 43 degrees around some avidly metabolizing tissues. Let's take a spot on this curve right here. I'm just going to go straight up from this little notch. So here you see that's the saturation of hemoglobin at that particular partial pressure at 37 degrees. Now we'll carry that blood to a hotter spot. Look how much the saturation is, way lower. What that means is, as we heat up conditions, oxygen comes off the hemoglobin. The affinity of hemoglobin decreases, or I think a better way to say that is probably that the oxygen has more energy and it breaks free. Everything has more energy at a higher temperature. The oxygens break loose, busting loose. And finally, the pH. In the tissues, the pH is more acidic. That's because we have more CO2 hanging around. It's a circled oxygen. We have more CO2 hanging around there because it's being produced by the cells, and we know that there's 36 or 45 or 46 millimeters of mercury partial pressure of CO2 there, and CO2 can form carbonic acid and then can dissociate into hydrogen ions and bicarbonate. We form hydrogen ions, acid. That drop in pH also reduces the affinity of hemoglobin for oxygen. So in this purple line again here, as we go into the tissues, it's the same effect. If we had a certain amount of saturation of oxygen on the hemoglobin at a particular partial pressure, and then we acidify things, we drop down. We have a lower saturation at that same partial pressure. So as we acidify things in the tissues, oxygen comes off the hemoglobin. So the amount of bisphosphoglycerate, or they wrote diphosphoglycerate, that's a sort of a frowned upon naming scheme for chemicals nowadays, that lowers the affinity of hemoglobin. Higher temperature lowers the affinity for hemoglobin. More acidic lowers the affinity for hemoglobin. So those three properties you should be able to tell me about that help us unload oxygen in the tissues, and it also helps us load the oxygen in the lungs. Look what's happening in the lungs. There's way less bisphosphoglycerate in the lungs and the red blood cells. There's a cooler temperature. We're bringing in cool air, and yes, we warm it in the nasal cavity, but it's still going to be cooler in the lungs than anywhere else in your body. We have a more alkaline pH in the lungs. So all of those three things help load oxygen in the lungs, unload oxygen in the tissues. Pretty amazing. Incidentally, that effect of the pH, the drop in pH on the affinity of hemoglobin for oxygen is called the Bohr effect. You should probably know that term if I ask you. All right. Quick aside, what about carbon monoxide? Terrible stuff, right? Carbon monoxide, hemoglobin has a very high affinity for carbon monoxide, much more so, 250 times that for oxygen. So if a little carbon dioxide gets in your blood, it clings onto the hemoglobin, and there's no place for oxygen to bind. So we'll, at only 0.2 to 0.4 millimeters partial pressure of carbon monoxide will saturate the hemoglobin, and there's no oxygen carrying capacity, and you die. Right? The ventilation doesn't even increase. Your body doesn't even respond because the partial pressure of oxygen is normal. It's just that none of it's bound to the hemoglobin. So we can't deliver any of it to the tissues, including the brain and the heart. Very bad situation, obviously. What can we do about it if it's not too late? One thing we can do is ventilate with pure oxygen. You drive up the partial pressure of oxygen, try to help displace some of that carbon monoxide, and eventually we will breathe it out. Little tiny bits of it at a time will be breathed out if we can keep the body going. We can also ventilate with some CO2 mixed in, because we're kind of getting ahead of ourselves here, but we said that the signal for ventilation is CO2. 40 millimeters of mercury partial pressure of CO2. That's the state of things when we're breathing in fresh air that has 0 CO2. If we breathe in some air that has a bunch of CO2 already, that's going to raise the alveolar CO2, raise the arterial CO2, and that's going to tell the chemoreceptors, hey man, we got to start ventilating more. We're not getting the job done. You can trick those chemoreceptors into driving up the ventilation rate and get things going and try to help get rid of that carbon monoxide from the blood. Okay, now we're going to talk about hypoxia. That's actually a type of hypoxia, having carbon monoxide blocking your hemoglobin from carrying oxygen. But let's talk about hypoxia more generally, and we'll look at some different categories of causes of hypoxia. Firstly, if you don't have enough oxygen carrying capacity of the blood, that's the definition I wanted you to learn for anemia. Not enough oxygen carrying capacity in the blood, then we have a type of hypoxia. As the blood is circulating, all of the hemoglobin is saturated with 4 oxygens per hemoglobin, but that's not enough because there's not enough hemoglobin. We have anemic hypoxia. Ischemic hypoxia means that we're not delivering enough oxygenated blood to the tissues, right? It's just not coming fast enough to meet the needs of the tissues, right? If you're in heart failure, your heart can't pump, it doesn't have enough cardiac output to deliver enough blood flow to all your tissues, and you're going to have ischemic hypoxia. Ischemia means not enough blood flow. If there's something wrong with your circulation, impairing the circulation, or an embolism. An embolism is what happens when a blood clot forms inside the vascular system, breaks loose, travels into the heart, and then out into an artery, and then into smaller and smaller arteries, and clogs and blocks flow. You're going to have ischemic hypoxia. Histotoxic hypoxia. This is what happens when we have excellent oxygenation of the hemoglobin, excellent amounts of hemoglobin. We send the oxygen around to the tissues, but they can't use the oxygen. Cyanide poisoning, it blocks the electron transport chains in the mitochondria, and that's where we use the oxygen. Right? We can't do aerobic respiration. Again, you're going to die because the heart cannot do anaerobic respiration. It must do aerobic respiration. If we stick enough cyanide in there, then it won't be possible. The cells can't use the oxygen. So that's called histotoxic. Histo means having to do with cells. Toxic, hypoxia. Finally, hypoxemic hypoxia. Hypoxemic means we don't have enough oxygen in the blood. It's not saturated. In fact, people with hypoxemic hypoxia appear blue in color because deoxyhemoglobin has a different color than oxyhemoglobin. Oxyhemoglobin is a brighter red color when it's saturated. Deoxyhemoglobin has more of a bluish tint, and we can recognize that. We're very good at, our eyes are extremely good at observing differences in color shades. Why would someone have hypoxemic hypoxia? That's because a number of things, but one of them is carbon monoxide, we just said. If there's carbon monoxide on all the hemoglobin, then we can't get enough oxygen into the blood, so the blood is deficient in oxygen. We have hypoxemic hypoxia. Excuse me. What if we have emphysema? We can't ventilate adequately. We have low ventilation and high perfusion everywhere in the lung because we just can't ventilate well. That's called a ventilation perfusion mismatch, or a VQ mismatch, and when it happens in this way, it's called a shunt. Blood is shunting through the lung without getting oxygenated. So that's a big problem in that disease. We don't have enough surface area to exchange gas, and we can't ventilate well enough because we can't get the air out, and we have this bad VQ problem. Hypoxemic hypoxia. What if you have pneumonia? You have fluid all in the lungs, and we have hypoxemic hypoxia. What if you have congestive heart failure? We have a ton of edema in the lungs. Hypoxemic hypoxia, blood that is not adequately oxygenated. What if you go up on top of a high mountain where the partial pressure of oxygen is super low? Above 10,000 feet elevation, things really get a little bit difficult for the respiratory system because the atmospheric oxygen is so low. Hypoxemic hypoxia. All right, quickly, let's look at CO2 transport before we call it a day. CO2 is transported in a very different way from hemoglobin. 3 different ways that CO2 is transported in the blood. First of all, there's a significant amount of CO2 that dissolves in the blood, in the plasma. Because it's so much more soluble, oxygen doesn't dissolve to an appreciable degree. The amount of dissolved oxygen is meaningless or minimal in the blood. It's all got to be carried on hemoglobin. But CO2, we can stick quite a bit of it into the blood here. 20% of the CO2 can bind to hemoglobin. But interestingly, it's not binding at the heme ions. It's not competing with oxygen for the sites. It's binding elsewhere on the hemoglobin, on the globin chains. That is a cool thing right there. And what's even more surprising and just fascinating is that as CO2 binds, as oxygen is dissociated, CO2 binds with higher avidity. It's not binding near the oxygen, but it's another allosteric effect. As oxygen unloads, CO2 binds to the hemoglobin better. What's happening in the tissues? We're unloading CO2, and now the hemoglobin starts we dissociate O2, and the hemoglobin starts mopping up CO2. That's called the Haldane effect. Another genius-level scientist who contributed that little tidbit to our knowledge of gas transport. 2/3, though, of the CO2 is transported as bicarbonate ion. Whoa, what? So what happens is the CO2 is formed in the tissues, diffuses into the red blood cell, and in the red blood cell is some carbonic anhydrase enzyme, and it can combine CO2 plus water to form carbonic acid. Got to have that enzyme there, or this doesn't really happen to any appreciable degree. Once we form carbonic acid, it dissociates to form hydrogen ions and bicarbonate ions, right? So that same carbon right here that we had to start with, that came from the cells, is now over here in the form of bicarbonate ion, and the bicarbonate diffuses out into the blood in exchange for chloride ions. Negative charge going out, negative charge coming in. But all that bicarbonate goes into the blood plasma. My PowerPoint appears to be stuck. Into the blood plasma, and so we're going to carry that bicarbonate ion to the lungs, and that will allow us then to return things the other way. As we go to the lungs, CO2 is being breathed out, and as CO2 breathes out, we're going to, here we are in the lungs, CO2 is breathing out, we can bring that bicarbonate right back into the red blood cells from the plasma, form carbonic acid once again, carbonic anhydrase immediately splits them, and the CO2 can be breathed back out again. Form CO2 in the tissues, form bicarbonate ion, deliver it, transport it to the lungs, take that bicarbonate, form it back into carbonic acid, split it, and form CO2 once again, and breathe out that CO2. Pretty amazing. So we've got dissolved CO2, we've got CO2 bound to hemoglobin. Where do we have that? Do we have that anywhere? Oh yeah, HB hemoglobin, that's a nickname for hemoglobin, plus CO2 produces HbCO2, that's the hemoglobin bound to CO2, and then we have forming bicarbonate ions. So that's the way that CO2 is transported in the blood. And this is showing you, gee, if we form bicarbonate ion, we also form some hydrogen ions, we got to buffer that stuff. And it turns out that buffering happens because of the hemoglobin protein. There's so much protein in those red blood cells, it can mop up those hydrogen ions in the red blood cells. But it's still more acidic, and that helps drive off the oxygen. The Bohr effect, right? We said that it was more acidic in the tissues. Why? We have all this hydrogen ion coming from the dissociation of carbonic acid. So there's those hydrogen ions. As oxygen dissociates, it allows CO2 to bind, but also as hydrogen ions are produced, it drives the oxygen off the hemoglobin. So a wonderful balancing act of various types that allow us to deliver blood, deliver the gases we want. Okay, there's one last topic, and I had forgotten this topic was here. The respiratory quotient, what is that? When you think about oxidizing fuel molecules and producing ATP by aerobic respiration, oxygen is utilized as the electron acceptor from the electron transport chain, forming water molecules, and CO2s are produced by breaking off the carbons from our fuel molecules, glucose and fatty acids, and breathed out. And something you may not have thought about. What's the relationship between the number of moles of CO2 produced relative to the number of moles of oxygen utilized over a certain period of time? Well, it turns out that if you have pure carbohydrate metabolism, glucose as the entire sole source of ATP production by aerobic respiration, you'll have exactly a 1-to-1 correspondence. We'll have 1 mole of CO2 produced for every mole of oxygen utilized, and therefore the ratio is 1. The respiratory quotient is 1. If you could somehow have a situation where 100% fatty acids were being used for oxidation to get ATPs, the ratio would be 0.7, amazingly. It's not a 1-to-1 molar correspondence then. It's a whole different oxidative fuel source. Excuse me. At rest, it turns out, in your body, both things are happening simultaneously. You're using some fatty acids for energy, you're using some glucose, and so the respiratory quotient is 0.85. The reason to mention this is because it's a really cool thing. Just by looking at that number and knowing that the respiratory quotient for carb is 1 and for fatty acids is 0.7, we can figure out exactly what percent of fatty acids versus glucose is being utilized at any point in time for making energy in your body. That comes into play in understanding how your body responds to the demands of exercise, for example. Anyway, that's it for that section of the story. I know that was a little bit longish, and next time we'll get back together and look at some more aspects of the functioning of the respiratory system.